Okay. Good morning, students. In previous class or in previous video, we have discussed regarding poultry farm management, definition of poultry. After that, we have discussed about what are the components of poultry that can help us to means successful poultry management. Poultry management successful ಆಗಬೇಕಾದ್ರೆ ಯಾವ ಕಂಪೋನೆಂಟ್ ಬೇಕು ಅದರ ಬಗ್ಗೆ ನಾವು ಏನಂತಂದಾಗ yesterday ಕ್ಲಾಸ್ ನಾಗ ಡಿಸ್ಕಸ್ ಮಾಡಿದೀವಿ. ಆ ನಂತರ what we have discussed in previous class, in the sense we have discussed about the animal breeding. That is the very important concept regarding the animal husbandry. In animal breeding, what else we have discussed yesterday, in the sense we have discussed about the breed, definition of breed. Breed is nothing but a group of animals which are having the common and different characters, like what I told you, in the sense qualities. What are those qualities you are going to means we are discussed in a breed, in the sense their appearance, features, etc. That is about breed. After that, we have discussed about crossing or mating of these animals to get the desirable characters or increase the quality and productivity. Quality and productivity in case of milk or in case of meat, whatever it may be. So that's for that purpose we used to go for the mating in a under the concept what we are going to call it as a animal breeding. In that we have studied about the inbreeding and outbreeding. In inbreeding we are studied about the homozygosity, inbreeding depression. In outbreeding we are discussed the definition of outbreeding, and there are the two types of outbreeding we have studied. One is outcrossing and crossbreeding. Then we studied about interspecific hybridization, definition, example, and the important thing you are going to remember, that is the example for crossbreeding. In the sense Hisardile, that is the new breed of a sheep which is developed in Punjab by crossing Bikaneri and what I told you, Merino Rams. The combination of Bikaneri and Merino Rams, it gave result into the Hisardile, and that is the technique or the type of crossbreeding. That is the type of crossbreeding. These are all the things we have studied still for so far. Now, in today's class, what we are going to go for, in the sense, yesterday we have studied about the breeding, the kinds of breeding, in the sense inbreeding, outbreeding, in outbreeding, outcrossing, crossbreeding, then interspecific breeding, etc., etc. In the sense we are going to go for mating the animals in a breeding. So now in today's class, what we are going to study, in the sense controlled breeding experiment. We are going to study few experiments which can help us to control what we are going to call it as a breeding. When we are going to go for a breeding, whatever we need, how we need, and how much we need, in such a way that we are going to go for a breeding animals, and we are going to get the further whatever we are going to call that as a result. That's what in today's class we are going to discuss about controlled breeding experiment. Controlled breeding experiments. Controlled breeding experiments. Controlled breeding experiments. Okay. In controlled breeding experiments, we are going to study two things. One is artificial insemination. First one is we are going to study about artificial, ARTIFICIAL, artificial insemination, INSEMINATION, artificial insemination. In the sense, yesterday I told you about the breeding. In breeding, we have studied how many kinds of breeding. One is inbreeding, and second one is outbreeding. And we have studied again two kinds of or two types of outbreedings. What are those in the sense? Outcrossing and outbreeding. Outcrossing and crossbreeding. Outcrossing and crossbreeding. So these definitions we have studied yesterday regarding the breeding, and in today's class what we are going to discuss about controlled breeding experiment. Controlled breeding experiment. What are the experiments that can help us to what controlled breeding? In the sense that can means breeding it can happen under our control. In the sense human control. So first one is artificial insemination. Now artificial insemination, first you used to know the definition of semen. First you used to know the definition of semen. I think you all are aware about the semen. What is mean by semen? The semen word it came in a third chapter, human reproduction. Human reproduction kalimunda ನಾವು ಏನು ಕಲ್ತಿದ್ವಿ ಅಂತಂದಾಗ semen definition ಕಲ್ತಿದ್ವಿ. What is semen ಅಂತಂದಾಗ semen is a seminal plasma with sperm. With sperm. Seminal plasma with sperm. Seminal plasma with sperm is called as a semen. Now here artificial insemination, and you all what aware about the insemination. Insemination in the sense what? Release of sperms into the female genital tract. Release of sperm into the female genital tract. Naturally, that is what during the mating, that is what we are going to call that as a insemination. Now here in this particular concept, controlled breeding experiment, we are going to study artificial insemination in the sense what? Artificial in the sense by what we are going to call that as a artificially we are going to introduce the semen into the female genital tract, where we are going to introduce the semen. It is injected into the reproductive tract of a female. In the sense what? The semen injected to the female reproductive tract, or you can write here introduce. Semen can introduce into the female reproductive tract. In the sense, what is going to happen in an artificial insemination? We used to choose one what we are going to call as a superior male, and used to collect the semen. Then these semens are introduced into the female reproductive tract. Understood? And these semens, when you are going to collect, immediately collecting the semen, you can introduce into the female genital tract or female reproductive tract, or else you can preserve. Means here in artificial insemination, what they used to do? They used to preserve. The semen may be used immediately or it can be frozen. Semen it can be used immediately or it can be frozen. In the sense, what we are going to call that one as a that is going to kept under the what we are going to call that as a minus 196 degrees centigrade. In the sense that can be frozen, and after that the frozen semen it will be used after some days, after few days. Means here in artificial insemination, what we used to do? Artificial insemination is nothing but the collection of a semen from the superior male, or which male we are interested. In the sense which kind of male we are interested to get the offspring. We are going to collect the semen from that particular superior male. Then we are going to introduce the semen into the female reproductive tract. That is what artificial insemination. And while doing the artificial insemination, you can collect the semen and immediately you can introduce into the female reproductive tract, or else they used to frozen, in the sense first used to collect the semen, used to frozen those and stored for a few days, and when we are required, that time we are going to go for introducing into the female reproductive tract. Understood? So in this way we are going to carry out the artificial insemination under the what controlled breeding experiment. This is the first experiment. Okay, and in this experiment we used to carry out desirable mating. We used to carry out desirable mating. Desirable mating in the sense what? From which superior male we want to means we want semen. Those semen only we used to collect, and we used to get the offsprings like that particular whatever we are selected the male. Understood? In the sense, here the artificial insemination helps to desirable mating, and artificial insemination helps to overcome several normal matings. Several normal mating. And during the normal mating, what is going to happen? Sometime during normal mating, what is going to happen? If the sperm count is low, that time the conciseness it is going to fail. And here in artificial insemination, we used to introduce the particular numbers and healthy sperms into the female reproductive tract. That is help us to overcome what we are going to call that one as a normal, several problems during the normal mating. So understood? So this is about what we are going to call that one as the first experiment for the controlled breeding experiment, that is artificial insemination. In artificial insemination, what we are going to means first what you used to know about the semen. Semen is nothing but the seminal plasma with the sperm. This semen, what in artificial insemination, semen is collected from what we are going to call as a superior male, and then it is introduced into female reproductive tract. That is what we are going to call that one as a artificial insemination, and this can be done immediately after collecting the semen, or else semens are going to frozen and they used after after some days when we required. And in frozen condition also we can introduce the what semen into the female reproductive tract, and this can what we are going to call that as this can help us to get the desirable mating, and this can help us to overcome several problems which are there during the normal mating. Some problems which are there during the normal mating, by them we used to overcome by using the artificial insemination. This is about the first what we are going to call as a experiment under the controlled breeding experiment, that is artificial insemination. And now I am going to discuss one important what we are going to call that one as a experiment which is help us in a breeding, that is what the short form MOET, and this question is asked so many times for 1 mark. What is the long form of MOET? MOET is nothing but multiple ovulation embryo transfer. Multiple ovulation embryo transfer. Multiple ovulation embryo transfer technology. Multiple ovulation embryo transfer technology. This is what 1 mark question. So many times this question is appeared in your final exam. What is MOET? MOET is nothing but multiple ovulation embryo transfer technology. For this we are going to get 1 mark. Understood? Now here this technique is used for herd improvement. This technique is used for herd improvement, and this is very important word. I used to tell you herd improvement. This is very important word, and that can means MOET is helps to improve or improvement in a herd. Herd in the sense what? Multiple numbers or numbers in the sense more than 3 or 4 like that. Here herd in the sense you can use this word for a sheep herd. Sheep herd in the sense so many sheep. Cow herd in the sense so many number of cow. The numbers, a group of in a group number that can be what we are going to call as a explained with this word herd. Herd improvement in the sense here the MOET is used for herd improvement in the sense at a time many ovulation we can we used to go for the many ovulation. In the sense in cow what is going to happen, cow atandage ಆಕಳ ನೋಡಿರಬಹುದು ಅಥವಾ ಬಫೆಲೋಸ್ ನೋಡಿರಬಹುದು, at a time a single what we are going to call that as a ovulation is to take place. Ovulation in the sense what is the ovulation? The secondary oocyte, the rupture of Graafian follicle and the release of the secondary oocyte is called as a ovulation. This you are studied again in a third chapter. So these definitions are very important. Okay, so ovulation, what is going to happen regarding the cow in the sense at a time a single means single secondary oocyte is going to release. But in this particular multiple ovulation in the sense at a time we are going to go for multiple ovulation by using the technique. That is what we are going to means the technique name itself. What multiple ovulation embryo transfer, and that is help for herd improvement in the sense what at a time what we are going to call that as a we are going to call ovulation of one egg. Instead of one egg we used to make that ovulation of many eggs in the sense herd improvement. Many, understood? That is the herd improvement. And this question is asked I think in 2019 question paper for 3 months, and there that fellow is asked this question every time what is going to happen. ಎಲ್ಲ ಕ್ವಶನ್ ಪೇಪರ್ ನಾಗ ಹೆಂಗೆ ಕ್ವಶನ್ ಕೇಳೋಣ ಅಂತಂದಾಗ ಭಾಳ ಸತಿ ಕ್ವಶನ್ ಬಂದೈತಿ. ಅಗೇನ್ ಕೇಳೋಣ ವಾಟ್ ಈಸ್ MOET and what are the what we are going to call as steps involved in a multiple ovulation technique ಅಂತ ಕೇಳೋಣ. ಬಟ್ 2019 ನಾಗ ಏನು ಮಾಡ್ಯಾನಪ್ಪ ಅಂತಂದಾಗ in 2019 he asked the question using this particular word herd improvement. Explain the steps involved in herd improvement technology. At that time so many students they are got confused. ಯಾಕಂತಂದಾಗ ಯಾವುದೇ ಕ್ಲಾಸ್ನಾಗ ಅಥವಾ ಎಲ್ಲಿ ಕೂಡ ಇದನ್ನ ಕರೆಕ್ಟ್ ಆಗಿ ಎಲ್ಲ ಹೇಳ್ತಾ ಇದ್ನ ಮಲ್ಟಿಪಲ್ ಓವ್ಯುಲೇಷನ್ ಎಂಬ್ರಿಯೋ ಟ್ರಾನ್ಸ್ಫರ್ ಅಂತಂದ ನಂತರ ಒಂದು 3 ಸ್ಟೆಪ್ಸ್ ಅದಾವ ವಾಟ್ ಯೂಸ್ ಟು ಗೋ ಫಾರ್ ಮೀನ್ಸ್ ಫಾರ್ ಡೂಯಿಂಗ್ ದಿ ಮಲ್ಟಿಪಲ್ ಓವ್ಯುಲೇಷನ್ ಎಂಬ್ರಿಯೋ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡೋದು ಏನೇನು ಸ್ಟೆಪ್ಸ್ ನೀವು ತಗೋಬೇಕಲ್ಲ ಅವು ಈಸಿ ಸ್ಟೆಪ್ಸ್ ಅದಾವ. ಆದ್ರೆ ಸ್ಟೂಡೆಂಟ್ಸ್ ಏನಾಯ್ತಪ್ಪ ಅಂತಂದಾಗ ಡೈರೆಕ್ಟ್ ಕ್ವಶನ್ ಕೇಳಿದಾಗ ಮಾತ್ರ ಬರೋದು. indirect ಅಂದಾಗ ಓನ್ಲಿ he use the herd improvement technique. Explain the herd improvement technique. Okay, into question ಕೇಳಿದ್ರೆ 3 ಮಾರ್ಕ್ಸ್ ಕ್ವಶನ್ ಕೇಳೋದು. So that time so many students they left the question, they are not understood what you used to write. If the question is asked directly then they are able to write. So ಇದು ಕರೆಕ್ಟ್ ಆಗಿ ನೆನಪಿಟ್ಕೋಬೇಕು ನೀವು ಏನಂತಂದಾಗ herd improvement. Okay, now I am going to start explaining about the what multiple ovulation embryo transport technology. It is one of the such program for herd improvement. I told you already this is the one of a such program or experiment which is used for the herd improvement. One of the such what we are going to call as a program that is help for the herd improvement. And in this method what we used to do, how we are going to carry out the MOET, multiple embryo transfer. First we used to inject the hormone like FSH, for what we are going to call that as a superior female cow. What we used to do, cow is administered the hormone or inject the hormone like FSH for the what we are going to call as a superior cow. And you all know about the long form of FSH. I told you, and we have studied about the FSH also in a third chapter, that is the human reproduction. In human reproduction, we have studied about the FSH. FSH is nothing but follicular stimulating hormone. Now, what is the function of this FSH in female? In the sense, it is helpful for folliculogenesis. It is helpful for folliculogenesis. In the sense, what is the folliculogenesis? The development of a follicles from primary to the Graafian follicle. That is what we are going to call as a folliculogenesis. And we have studied in humans. At that means we have studied at the last stage, embryo, in the sense Graafian follicle. When the Graafian follicle is going to mature, and that time the mature Graafian follicle is going to rupture, and the secondary oocyte is released from the mature Graafian follicle. That is what we are means that is called as a ovulation. Now here the FSH, when we are going to administer for the female cow, superior female cow, that time it induce what is going to happen? FSH induce the folliculogenesis. Folliculogenesis in the sense the development of follicles. Understood? Now, and follicular maturation, and it what we are going to call that one as a superior ovulation. FSH induces the folliculogenesis and superior ovulation. Folliculogenesis and superior ovulation. In the sense, here the ovulation is taken place. It takes place instead of one egg, many eggs are going to form there. Next, what is going to happen? I have written here superior ovulation. Next one is instead. What? Instead of one egg, normally instead of one egg per cycle, instead of one egg per cycle, we can get 6 to 8 eggs. We get 6 to 8 eggs in a per cycle. Due to what? The administration of FSH. What we are going to go for in multiple ovulation embryo transfer technology is to administrate or is to inject the hormone like FSH for the superior female cow, and this FSH induces the folliculogenesis and superior ovulation. That helps, instead of one egg per cycle, we get the 6 to 8 eggs per cycle, or instead of one egg per cycle, the cow produces 6 to 8 eggs. 6 to 8 eggs. And now what we are going to go for next? Next, after the production of eggs, the animal or a superior cow, the superior is mated. The superior cow is mated with elite bull or artificial insemination. Elite bull or artificial insemination or artificially inseminated. What we are going to means in MOET, after the production of 6 to 8 eggs per cycle, that cow is mated with the elite bull or artificially inseminated. Then what is going to happen after this? After the mating, fertilization of these eggs is to take place, and what is going to happen there? The fertilized eggs 8 to 32 celled stage. 8 to 32 celled stage are removed non-surgically. And introduced into surrogate mother. This is very important. Surrogate mother. SURROGATE. Surrogate mother. Understood? Introduced into the surrogate mother. The genetic mother is available for the superovulation. Then I will tell you now once again what is the particular MOET. MOET ಅಂದ್ರೆ ಏನ ಕನ್ನಡದಾಗ ನಾನು ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತೀನಿ. MOET ಅಂದ್ರೆ first we are going to select one superior cow, and that superior cow naturally what is going to happen per cycle it can produce the one egg. But here in MOET multiple ovulation embryo transfer ನಾವು ಏನು ಮಾಡ್ತೀವಿ, we are going to select the superior cow and we are going to administrate or inject the hormone like FSH for the superior cow, and this FSH induces the folliculogenesis and superovulation. Andre next point ಏನು ಬರ್ತೈತಿ, instead of one egg per cycle, this superior cow produce the 6 to 8 eggs. It can produce the 6 to 8 eggs. Okay, our superior cow get eggs going to 6 to 8. After that, what we are going to do is to go for mating that superior cow which has 6 to 8 eggs with elite bull. Elite bull in the sense ಒಂದು ಯಾವ ಪಾರ್ಟ್ನ ಸ್ಟ್ರಾಂಗರ್ ಅಥವಾ ಸುಪೀರಿಯರ್ ಮೇಲ್ ಬುಲ್ ಇರ್ತೈತಿ, ಅದರಿಂದ ನಾವೇನು ಮಾಡೋದು ಮೇಟಿಂಗ್ ಮಾಡಿಸೋದು. ಆರ್ ಎಲ್ಸ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಗೋ ಫಾರ್ ದಿ ಆರ್ಟಿಫಿಷಿಯಲ್ ಇನ್ಸೆಮಿನೇಷನ್. ನಾವು ಇದಕ್ಕಿಂತ ಮುಂಚೆ ಒಂದು ಎಕ್ಸ್ಪ್ಲೇನ್ ನೋಡಿದ್ದು, ಕಲೆಕ್ಷನ್ ಆಫ್ ಸೆಮೆನ್ ಅಂಡ್ ದೆನ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಇಂಟ್ರೊಡ್ಯೂಸ್ into the female reproductive tract of particular what we are going to call as a superior female cow. ಹಾಕಿದಾಗ ಏನಾಗ್ತೈತಿ, then the fertilization is going to take place. You all are aware about the fertilization in the sense the sperm union of a female gamete, male gamete and female gamete to produce the zygote. ಹಂಗಿಲ್ಲ ಏನಾಗ್ತೈತಿ, once you are going to introduce or mated with the elite bull or a artificial insemination, the eggs are fertilized. Fertilized by the sperms. Another fertilization ಏನಾಗ್ತೈತಿ ಅಂತಂದಾಗ what I told you, zygote, 2n number of zygote ಆಗ್ತೈತಿ, and this zygote start dividing. Okay, 2 cell stage, then 4 cell stage, 8 cell stage, 16 cell stage or 32 cell stage, like that it is going to start dividing, and after that it is going to form a blastula, gastrula. These all the things you have studied in third chapter again. Implantation also ನಾವು ಕಲ್ತಿದ್ದೀವಿ, ಅದೆಲ್ಲ ಕಲಿಯಂಗಿಲ್ಲ. ಏನಾಗ್ತೈತೆ ಅಂತ ಅದು ನಿಮಗೆ ಆಲ್ರೆಡಿ ಯು ನೋ ಅಬೌಟ್ ದ implantation and all, and what are the stages up to the what implantation, after the insemination, then fertilization, then 2 celled, 4 celled, 8 celled, 16, 32, 64, morula, then blastula, gastrula. These are all the things you have studied in a third chapter. Now here, once the fertilization is happened, how mated this superior female is mated with the elite bull or a artificial insemination, then zygote is going to produce, then it is going to start developing or dividing, and when it is come to the 8 to 32 cell stage, that time that can be removed non-surgically and introduced into the surrogate mother. This is very important. What is mean by surrogate mother? Surrogate mother in the sense the cows ಅಂದ್ರೆ ಹೆಂಗ ಈಗ ನೋಡ್ರಿ, ಈಗ I used to draw the diagram in such a way that can help you to understood what I am telling you. Understood? ಈಗ ಏನಂತಂದಾಗ surrogate mother ಅಂದ್ರೆ ಏನು? For example, I am going to draw here one diagram. This is what we are superior female cow we are selected. Then after that what we did, we are injected or administrated FSH for this cow, and in this cow we are going to get at a time, instead of one egg we are going to get 1 2 3 4 5 6 to 8 eggs. Then what we are going to go for, we used to make this superior cow with a elite bull or artificial insemination. Another ಏನಕ್ಕೆ fertilization, and after that ಇದೇನ್ ಸ್ಟೇಜ್ ಇಲ್ಲ 8 to 32 cell stage banana. Surgically we are going to remove the eggs from this superior female, and then we are going to introduce in such a way that one egg to one surrogate mother, another egg to another surrogate mother, 32 cell, another one, another, like this. At a time we are going to get how many? 5 to 6 or a 6 to 8 conceived cows, and these cows we are going to call as a surrogate mother. Here these cows are not genetically what related to these eggs, because these eggs are taken from the superior female ದಿಂದ ನಾವು egg ತಗೊಂಡಿರ್ತೀವಿ, ಆ ಒಂದು ಏನಪ್ಪ surrogate mother ಹಾಕ್ತೀವಿ. ಹಾಗಾಗಿ at a time ಒಂದೇ ಕಾವು ಏನಪ್ಪ ಅಂತಂದ ಕನ್ಸೀವ್ ಅಥವಾ ಒಂದೇ ಕಾವು ಪ್ರೆಗ್ನೆಂಟ್ ಆಗಿ ಅದರಿಂದ ಕರು ಹಾಕಿ ಅದರಿಂದ ನಾವು ಮಿಲ್ಕ್ ತಗೋತೀವಿ. ಬಟ್ ಇಲ್ಲಿ ಏನಾಗ್ತೈತೆ ಅಂತಂದಾಗ at a time ನಾವು 6 ಒಂದು ಕಾವು ಏನಪ್ಪ ಅಂತಂದಾಗ ಕನ್ಸೀವ್ ಮಾಡಿ 6 ಕಾವು at a time ಕರು ಹಾಕಿ 6 ಕಾವುದಿಂದ ಏನಪ್ಪ ಅಂತಂದಾಗ ನಾವು ಮಿಲ್ಕ್ ಪ್ರೊಡಕ್ಷನ್ ಮಾಡಬಹುದು. ದಟ್ ಈಸ್ ವಾಟ್ ಹರ್ಡ್ ಇಂಪ್ರೂವ್ಮೆಂಟ್. Understood? That is what herd improvement. And once you are going to remove these 8 to 32 cell stage eggs, means what we are going to call it as a fertilizer eggs. After that again it is ready for next cycle. The genetical cow, superior cow is ready for the next cycle in such a way that this multiple ovulation embryo transfer can help us to the increase, means improve the herd improvement. Okay, and this is used in cattle, sheep, rabbit, buffalo, mares, etc. And this can help us to increase the high milk yielding breeds of female and high quality of what we are going to call it as a lean meat. We are going to get the lean meat with the less lipid. Okay, understood? This is about what the experiment we are studied in today's class. First one is artificial insemination, and second one is the what? Multiple ovulation embryo transfer. Thank you.